Welcome and good morning, good afternoon, and good evening. It is very, very exciting to have participants from around the world, both sharing their thoughts and participating in this webinar. I'm Dr. Lindsey Baden. I'm an infectious disease physician and investigator in Boston, a professor of medicine at Harvard Medical School. I work at Brigham and Women's Hospital, and I do vaccine development. I don't tell this group the importance of vaccines and their power to change health. The world now needs to be reminded how 50, 70 years ago, vaccine preventable diseases were dominant and were the leading causes of illness, morbidity, mortality across all parts of the world. The need for greater prevention for HIV, it continues to spread way too fast with millions of new infections per year. We need to use all of our prevention modalities, emerging PrEP with new formulations of medications that can be delivered in a way that are tolerable and expandable. We need vaccines because they are sustainable and they're economically favorable as we think about global deployment to all who could benefit. The science of HIV vaccine development has been quite remarkable, especially in the last decade. The path to be able to induce broadly neutralizing antibodies, something we have seen in long-term non-progressors that are able to control the virus and able to control a broad array of genetic variants of HIV, has illuminated a path to create these antibodies passively and, even more important, actively through vaccine elicited immune responses. We have a terrific group of speakers today with both global and scientific breadth and depth in their understanding of HIV, the need for vaccines, and the approach to be able to develop them. A procedural note: for those who have questions, please place your questions in the Q&A tab so that we can address them between talks and at the end. Let me turn this over to my co-moderator, Dr. Mgodi.
Thank you so much, Dr. Baden, and thank you for the great preamble. My name is Nyaradzo Mgodi. I'm Dr. Nyaradzo Mgodi from the University of Zimbabwe's Clinical Trials Research Center. I have almost 2 decades experience in conducting clinical trials for biomedical HIV prevention interventions spanning from oral PrEP, systemic PrEP, topical vaginal microbicides, and more recently, I've started working on broadly neutralizing antibodies as well as vaccines. So I'm really honored to be co-moderating this symposium with you, Lindsey, and also to welcome our panel of speakers and all our participants. So without much ado, perhaps just to remind people in terms of what is the mission of the ISV. So the mission is to encourage, establish, and promote the development and the use of vaccines or immunity to prevent and control infectious and non-infectious diseases in humans and animals. Also to develop a professional and academic society devoted to all aspects of vaccines, and to support and promote the career development of scientists in the field of vaccinology, including trainees. So these symposia are held quarterly, and this is the 3rd symposium. The last one was in April on RSV vaccines, and obviously this one is on HIV vaccines. The 4th and last mini-symposium will be in September on influenza vaccines. So please stay tuned for the next and last fourth mini-symposium. So as Lindsey mentioned, we've got a panel of interesting speakers, and our first speaker is Glenda Gray. Glenda has got a long bio, so I'll try to summarize. Glenda is she was the first female president and CEO of the South African Medical Research Council, and she's currently the Chief Scientific Officer at the SAMRC. She trained as a medical doctor and pediatrician at the University of the Witwatersrand, and she also co-founded and led the internationally renowned PHRU, a research unit based at the Chris Hani Baragwana Hospital in Soweto. She's internationally known for her research in HIV vaccines, and she's the co-PI of the HVTN, the HIV Vaccine Trials Network, and she is also the lead in the Brilliant Consortium for vaccines. She's received multiple awards, including South Africa's highest honor, the Order of Mapungubwe, which was awarded in 2013. In 2017, she was listed amongst the Times 100 influential people in the world, and in 2020, she was listed in the Forbes Top 50 Women in Africa. Her focus now is on the capacity development in science, with a special focus on transformation and increasing diversity in science. And I'm proud to say I'm one of her mentees, albeit maybe from afar, but South Africa, Zimbabwe, where we share borders. So Glenda, you've got 25 minutes for your presentation, and we'll have 5 minutes for Q&A at the end of this. And may I remind all participants to post your questions in the chat box. Over to you, Glenda. Thank you.
Thanks very much. I'm going to try and share my screen. Just tell me if you can see it.
Not yet. Yes, yes.
Okay. And now I'm just trying to put it onto, it worked very well when I started, and now let's just get it onto full screen now. Can you see it now?
It's coming. You're still not yet in presenter mode, but perfect.
And
I
now,
do see. Yes. Thank
am
you.
I now in presenter mode? Thanks.
Yes.
Thanks very much, Nyaradzo, my sister from Africa. It's lovely to be here with you. And I'm going to set the scene with about 20 slides and 2 messages about why we need an HIV vaccine. So I want to make the point that the world is not on track in controlling HIV. At a global AIDS update called AIDS at the Crossroads, the emphasis was made that there are inequalities that drive the pandemic, which have not been addressed. We know that there's a lack of progress on prevention, and the global numbers of new HIV infections are not declining fast enough. In 3 regions of the world, the numbers of HIV infections are increasing, and almost 1/4 of people living with HIV are not receiving HEART. 1 person dies from AIDS-related causes every minute. So we are at a crossroads, and whether the world ends AIDS depends on the path that we take. The path to end AIDS is not a mystery, it is a political and financial choice. This slide just shows you two important slides that shows you the number of HIV infections over a 20-year period, and shows you that that although there's a decline in the incidence of HIV, we still have not met our 2025 target, which is this year. If you have a look at the bottom part of the slide, you will see also that we are abysmally poor at offering pre-exposure prophylaxis in every region of the world. And here you see the red dots are where we're supposed to be, and the blue bars are where we are. And you can see across the board, we are lacking in the number of people who are using PrEP, albeit East and Southern Africa are the regions of the world hardest hit by HIV, who are also the biggest users of antiretroviral therapy. If you have a look at this slide, I think you've seen it before. It's where we're supposed to be, the trajectory, and where we are at the moment. And we do need to boost our toolkit to try and make sure we can reach the 2030 goal where we have HIV under control. I've spoke to you a little bit about prevention, but I can show you we can't treat ourselves out of the epidemic as well. Here we have the number of people receiving antiretroviral therapy, and here you can see our target. On the right is the bars of people living with HIV who know their status, and you can see above is the 2025 target. We're below that. If you have a look at people who know their HIV status and on treatment, we're below that. And people who are living with HIV on treatment who have suppressed, we are below that. So if we have people who are not diagnosed, not on treatment, and are still viremic, they are able to continue the onward transmission of HIV. There is also a disconnect between the amount of resources allocated to where the infections happen and the money we get. And so you can see in the blue bars is a number of new infections from among all key populations. And the red bar is the number of total HIV spending on prevention amongst people in key populations. So doesn't take a rocket scientist to see why we're off track and why we cannot make HIV a controllable disease because we're not spending the money where it needs to happen. Here is quite a sobering slide that shows you where we are. So where we are and where we should be. And so our target for to reduce new infections to under 370,000, and we are about just over 1 million. Reducing infections amongst young women, we're far below that. We're far below the amount of people getting effective antiretroviral therapy. We're far below the amount of people who need PrEP. We're on 3.5 million, where we need to get to 21 million. And so you can see whatever target we see, we are so far behind in managing HIV at a global level, including pediatric HIV, where we aim to get 75% of children living with HIV to have suppressed viral loads. We are only on 48%. And where we aim to get women who are living with HIV and who are pregnant and breastfeeding at 100% on treatment, we're only at 84%. So you can see that we're far below our ability to manage and to treat and prevent HIV. So I asked ChatGPT about why we still need an HIV vaccine. So ChatGPT told me that prevention is better than treatment. A vaccine would reduce global dependency on costly lifelong care and probably costly lifelong prevention. The global pandemic continues with 1 million new infections per annum. There are limitations of existing prevention tools. They cost a lot and they require regular interface into the healthcare system, whereas a vaccine would be more durable and more cost-effective. A vaccine would also protect vulnerable populations and provide equitable, stigma-free prevention. And I love that word, equitable, stigma-free prevention. And the scientific and public health breakthrough would represent a huge advance in immunology and virology and help us guide vaccine development for other more complex diseases. So ChatGPT tells me we need an HIV vaccine to stop the epidemic at its source, to provide long-term accessible protection, to reduce new infections globally, and to move from control to elimination. So what kind of immune responses are we required for an effective HIV vaccine? We want potent neutralizing antibody responses. They must be broadly reactive. They must be persistent. We need good memory T cells that suppress viral replication. And we need immune response that prevents viral escape. And it takes about 50 to 200 times more neutralizing antibodies to inhibit HIV than it does SARS-CoV-2. And also the rapid integration of HIV after acquisition and the inability for early therapy to interrupt this integration means that the goal of an HIV vaccine must be to prevent acquisition or eradicate recent infection. So can we use an HIV vaccine approach for both prevention and cure? Can we look at the work that's coming out? Peter will speak about the correlates of protection. Bill will talk about neutralizing antibodies. And there is work that shows that non-human primate work could help inform prevention and control. And so the reason why an HIV vaccine is important because it could work in both prevention and therapeutics. So there are several possible vaccine effects. One, we could try to prevent HIV acquisition altogether. We could try to prevent HIV becoming a systemic infection. And we can try prevent HIV from going from an infection to disease. So these are the possible strategies that one could get from an HIV vaccine. You'll hear later on about non-neutralizing approaches to prevent HIV based on the RB144 and the role of the V2 antibodies. I'm not going to talk about anything about the T-cell based vaccines, but these will be important to induce good CD8 T-cell responses. And Bill Schief is going to talk about vaccines that induce broadly neutralizing antibodies. I will just mention a little bit about immunoprophylaxis with BNABs that are attempting to do the same thing that Bill is doing as well. So in terms of the vaccine R&D pipeline, I want to acknowledge AVAC for this. And of the trials that we've done for efficacy, there's only been 2 studies that have been shown any evidence of efficaciousness, and that is the RB14 study conducted in Thailand that showed modest efficacy, and the AMP study, which showed that a monoclonal antibody with aimed at the CD4 binding site could, in virus sensitive, could protect against acquisition. So my first message is that there have been 3 non-neutralizing HIV vaccine efficacy studies that indicate that the V1, V2 loop antibodies may play a role in protection. And we can't yet eliminate this hypothesis because neither the 702 or the 705 regimen that we conducted achieved high enough titers to demonstrate this role. And hopefully Shan, at the end of this seminar, will talk about his work in this area. I'm going to quickly talk about the phase 2 proof of concept that looked at a monoclonal antibody, VRC01, that was aimed at the CD4 binding site. This was a large trial conducted both under Nyaradzi's leadership in Africa and one in Americas and in South America and in the Americas. And here you can see that this was a 3-arm study, high dose VRC01, low dose VRC01 and placebo. And here you can see that there was protective efficacy where there were sensitive viruses, and this was around 60 to 80% after 2 doses. And that if you weren't sensitive to this virus, then the protective efficacy was 0 by 80 weeks. But this was an important thing. This is my second message, is that this provided an important roadmap for preventing HIV acquisition, and for people like Bill Schief provides that roadmap for vaccine development. We believe that BNABs can be used for preventing breast milk transmission of HIV. The target neutralization titer will be a useful correlate in the work that people like Bill Schief are going to do. And AMP also demonstrated that we need to make BNABs to more than one site in order to be efficacious. So there are diverse efforts to achieve an HIV vaccine, and everything from community engagement to integrated analytics. We need the immunology, we need the virology, and we need the host genetics. We need to do experimental medicine studies that Bill will talk about, and we need to then take our best candidates back into human efficacy studies. And we have to do this over and over again to try and make sure that we can find an HIV vaccine that's efficacious. This is critical, the need for ongoing evaluation, discovery, and moving on into humans. And so this is the world that I want to see. A vaccine that's efficacious with a target product profile that prevents HIV infection amongst seronegative adults, a 50% efficacious vaccine at 24 months after first administration, well tolerated and safe, needs 2 months from first administration for a boost, and maybe we only need to do hopefully less than 5 doses administered over 12 months. So I just want to thank the HVTN and many funders who believe in moving an HIV vaccine program forward, and all the funders that have helped us to get this far at a global level. There are a lot of people at the HVTN that I want to acknowledge, including Larry and Lindsey and Nyaradzo and all the amazing people that without we would not be able to get this far. And I want to thank also, of course, our global CABs and all our participants in our clinical trials. So I'll stop sharing there and take any questions. Or I can ask ChatGPT to answer the questions.
Please go ahead, Lindsey.
No, for our, it is terrific to see hundreds of participants. So please put your questions and you will start with sort of the first question. The terrific as always in framing the scope and magnitude of the problem. What do you see as the biggest barrier to developing an HIV vaccine?
Money, I see as the biggest barrier. I think we have amazing scientists, and I think we need effort to iterate. We need to be able to get into humans as fast as possible, and we need good vaccine platforms that can help us. So I think we need a lot of political will and financial muster, and a process that fails fast. So maybe Bill from industry can talk about the concept of failing fast. And that is why discovery medicine trials are important, so that we can get into humans and fail fast and get back for translation. So I think it's a global endeavor that requires financial and political commitment to help the scientists move the needle, Lindsey. That's what I believe.
And just to, since words are charged, when you say fail fast, I think what you're saying is we learn a immunogen that does not create a protective vaccine, but shows us important biology may fail as a vaccine, but be incredibly informative scientifically.
It's very critical. And so what we like about failing fast is understanding why we failed and improving our approach. And so I think it's about being nimble and agile and moving as fast as we can, and then iterating using the clinical trials as the step to move forward, and not non-human primates. So I would like to see more clinical research and a rapid iteration back into the clinic. I think with new tools like AlphaFold and AI and machine learning, and the ability to visualize both the virus and the antibodies we make and the target sites, it makes us much more informed in how to develop and drive a vaccine program forward.
So thank you very much, Glenda. I'm looking at the chat. I don't see a question, but I've got a question of my own. So we've all gone through the agony over the past almost 2 decades with over, you showed, I think, more than 12 clinical trials where we only had 2 positive signals from the RV144 and from AMP. So, and there've been various strategies in terms of what should we do. Could you maybe talk a bit more about germline targeting, about targeting the V1, V2 loop, and also for some of our listeners here, participants here who might not know, if you could also take us through your work, our work in the Brilliant Consortium. Thank you, Glenda.
I think that we've made tremendous progress. So even though we asked specific hypotheses in our efficacy studies, and we came up with a definite answer. I think that we'll have Peter Gilbert that will talk a little bit more about the correlates and why correlates of protection are very important in trials that have failed, and to try and understand in subgroups whether there may be areas where a subgroup of participants did better than others. So I think that's very critical to analyze and understand the mechanisms of protection and why we never reached that. So I think that's important. So even though we've done these vaccine trials that have not resulted in an efficacious product, I believe that we contributed massively to the understanding of science. Bill will talk a little bit about the germline targeting, but the whole idea is from the AMP study, identifying that a target against the CD4 binding site could lead to protection, and the importance of us looking at these epitopes like V1, V2, like EMPER, like fusion protein, to try and develop immunogens to these target sites on the envelope, and see whether we could find a way, either through germline targeting or other strategies, to induce the broadly neutralizing antibodies that we saw with AMP. AMP's going to be the next step from AMP's going to be very important. Remember, we had a monoclonal antibody only to the CD4 binding site. By adding more monoclonal antibodies aimed at different sites of the envelope will help us in active immunization, in the translation. So if we can show protection, it will guide the work that Bill's doing, and to make sure that we can find ways of doing germline targeting or lineage-based vaccine approaches using multiple epitopes and not just one single target at a time. And hopefully Bill will talk about that going forward. So in terms of the Brilliant Consortium, we are hobbling along. We did have a big USAID grant that was focused on getting African scientists to contribute to developing an HIV vaccine. Unfortunately, USAID terminated our grant at the end of January, but we are working together with other people like the Gates Foundation to pick up some of the work that we did, and to collaborate going forward with other vaccine developers. The Brilliant Consortium, as you know, Nyaradzo, you were a member. We are an agile, nimble clinical trial consortium that can respond in a very fast way to do vaccine trials in Africa, experimental vaccine trials in Africa, supported by local clinical data management systems and local laboratories. So we do believe that we have in the future a big contribution to make to experimental medicine, as well as other stages of HIV vaccine design.
Terrific. And many questions are now coming in. Several of our audience members have raised the issue of vaccine hesitancy. How do we think about that and the implications to developing an HIV vaccine?
I think that's a great question, Lindsey, given the environment that we're living in. And I think it's important, that's why we bring our communities. So I stressed in my last slide the importance of co-creation, co-design, collaboration with the communities that are severely affected by HIV. And by having good partnerships, good community engagements, we can address the issue of vaccine hesitancy. And so I think it's a very important thing to make sure that when we are engaging with our communities, when we are designing our studies, we're raising the issue around the concerns of people and their issues with mRNA platforms, as well as the side effects that they are concerned about. But we can only do this, we can only move forward if we completely engage with our communities and they co-lead HIV vaccine trials with us in our communities.
So I'm watching the clock. You've got a bit of time, Glenda. So I'm just picking the questions at random, and I'll pose 2 questions. And this was bound to come in the advent of highly efficacious 6-monthly lenacapavir. Do we still need a vaccine, and how should future vaccine trials be designed ethically? And why is it that after 40 years into the epidemic, we still don't have an efficacious vaccine? I think you talked a bit about that, but if I've combined 2 questions there. Thank you.
Yeah, I mean, so I think in the last few days in the New York Times and in a couple of global literature, we've seen the failure to roll out a highly efficacious long-acting antiretroviral injection because of price, because of slowness in tech transfer, and in slowness in having generic equivalents. So that is the biggest problem, is around the cost. To deliver long-acting antiretrovirals to a woman in South Africa would cost R1 million a year. And quite frankly, no one can afford, our health systems can't afford R1 million a year for maybe 20 million people that need it in South Africa. So unless the prices drop down to less than $50 per dose, we'll never be able to rise to that occasion. The other issue around the long-acting antiretrovirals, which are very important, and I do wish they were cheap so that we could scale them up. But the issue is around the medicalization of HIV prevention. With oral PrEP, we managed to do this at a community level by having ongoing long-acting antiretrovirals. We re-medicalize HIV prevention, and which also may lead to lots of issues around stigma as we try and make sure the most targeted populations are brought in. So I think it's an important and powerful intervention, but it's only important and powerful if it's available. And at this moment in time, unfortunately, it's not available. So basically, we have market failure when it comes to long-acting antiretrovirals because of the cost. And that's just the bottom line, is that if we can't afford this kind of high cost for prevention in a poor country. So that's the one issue. Why haven't we been able to find an HIV vaccine? Well, first of all, we don't have a good animal model that gives us a good marker. HIV is replicating all the time. All the areas all the targets that we could use are hidden, and we don't have anyone that's been readily cured from HIV that we can follow and understand what were the mechanisms that prevented the HIV that prevented infection. So we still it's a very complicated virus, and it's one of the biggest challenges that we have in order to try and unravel the HIV vaccine. We haven't done badly. We know where to go. There are other infectious diseases like TB. After hundreds of years, we still don't have an effective TB vaccine for adults, and already we know that some of the other pathogens that are hard, like chlamydia, syphilis, and gonorrhea, we still don't have vaccines for them. So the important thing about an HIV vaccine discovery program is that we understand so much more now than we did before. And if we didn't do all of this work, we wouldn't know where to go. And hopefully Bill and Peter and Shan will help us figure out the way forward.
Thank you, Glenda. May I kindly ask you to go to the chat and try to address as many questions as you can, because they now flooding in. So thank you so much, Glenda. Lindsey, over to you.
Thank you, Dr. Mgodi. And thank you, Glenda, for a terrific overview of the tremendous infection force globally of HIV and the urgency for us to address this. Some of the questions in the Q&A we will ask subsequent speakers, as they may better be able to address some of them. Our next speaker, Bill, you can turn on your camera and microphone. William Schief is Vice President of Protein Design at Moderna, Professor of Immunology Microbiology at Scripps, Executive Director of Vaccine Design at IAVI's Neutralizing Antibodies Center at Scripps. He has a BS of Applied Mathematics from Yale, a PhD in Physics from University of Washington. His work focuses on computation-guided and structure-based design of vaccines and therapeutics for a wide range of diseases. For HIV vaccine development, the topic of this mini-symposium, Bill is pursuing the strategy of germline targeting vaccine design with many collaborators. This strategy aims to induce broadly neutralizing antibodies through a series of vaccinations that activate the appropriate naive B-cell precursor and then facilitates maturation to BNAb. This needs to be guided through B-cell somatic hypermutation, affinity maturation, and much of the work of the immune system will go on in germinal centers, memory B cells, plasma cells, though we detect a lot in the serum. Bill has been a forward thinker in how do we target B-cell ontogeny to allow us to have the immune response that we wish to have. This has profound vaccine implications. However, he will focus his remarks today on BNAB induction. Bill, thank you for joining us.
Thanks for having me, Lindsey and Dr. Mgodi and the other organizers. I'm really happy to be here. Let me share my slides. And it's nice to follow.
You need to invert your screens or go to full screen.
Yeah, okay, let's see. Does that work? No? Is that good?
Yeah, no, I think you have to switch screens.
Okay. Sorry. Show this maybe. Is that good for you?
This can work. It's still in the same position, but we can see it.
That means you're seeing the presenter view? Sorry.
Yes.
Oh, okay, sorry. Let's see if I can fix this. Sorry, everybody. Maybe I should just go.
Oh, you can go. This is adequate.
Like this. Yeah, sorry. Okay. I'll make it. Is that better?
It is fine.
Okay. Okay, yeah, so I wanted to talk about it's nice to follow Glenda. She gave a really great overview for why we need an HIV vaccine and some comments about, efforts toward it. And I'll just tell you about our efforts and she said, fail fast in the clinic. I think of it, sure, if you need to learn, you need to iterate in human clinical trials for sure, for absolutely. We're hoping to succeed quickly and move to the next step because making an HIV vaccine is complicated and we think we need to do lots of phase 1 clinical trials to learn and iterate and develop an optimal and protective vaccine. Let me. Glenda mentioned broadly neutralizing antibodies, and that's what I'll
You don't see the next slide, Bill. We're still on the opening slide.
All right, is this okay? Does this work? I'm so sorry.
You can go out and in again. For some reason, it is showing your opening slide only.
Okay, let's see. Darn. Is this good? So opening slide, next slide. I don't know what yours.
We're seeing presenter view, opening slide only.
That's so weird. Okay.
You may want to stop sharing and then try to reshare.
Yeah, let me do that. I actually have never shared from, okay. Stop sharing. Sorry, everybody. Let's try it again. Let me give you my desktop. Let's just go with that. Then it might just.
Yes, now we see HIV broadly neutralizing BNAbs with the cartoon on the right side.
And I will play from the start and go to this one. Is that good?
Yes.
Okay, perfect. Sorry about
this. Perfectly.
So on the right, we have a representation of the HIV trimeric spike, and we want to elicit antibodies that block the spike and prevent HIV from infecting human CD4 T cells. And different classes of broadly neutralizing antibodies bind to different epitopes on the spike. Let me do this. Laser pointer. So HIV BNAbs neutralize diverse isolates. They can protect against infection in non-human primates and humans. As Glenda mentioned in the AMP trial, that's where the human data came from. Different classes of BNAbs bind different epitopes on HIV envelope, as shown in this image. We think that an HIV vaccine will likely need to induce multiple classes of BNAbs in order to achieve sufficient neutralization, potency, and breadth to protect against diverse isolates circulating among humans. And we also believe that broad T-cell immunity will likely be needed for an optimal vaccine. I just don't have time to talk about it today. Many people are working on that as well. We aim to induce BNAbs against 2 or more epitopes for complementary neutralization coverage, titers, and breadth. And we and our collaborators have recently published on all of the epitopes, including clinical studies. And today I'm just going to focus on the CD4 binding site and a little bit on trimer delivery in humans. But we take this very seriously, and we're putting tons of effort. And I agree with Glenda's comment that funding is a limitation. I mean, doing all this work and pushing all these things forward, including into the clinic, takes a lot of resources and will be needed to continue. What is required to induce protective broadly neutralizing antibodies? What we want from a vaccine is to induce long-lived plasma cells that secrete broadly neutralizing antibodies in the serum and do that durably for a long period, so that if you're exposed to HIV, those antibodies neutralize it and prevent infection of your cells. We think that the way to get there is to start from naive B cells, from rare naive B cells like this one in red, that have the genetics shared with bNAbs. They have the right genetics that would allow the B cell receptor on this naive B cell to, if it gains somatic hypermutation, if it gains the right mutations, would allow this antibody to turn into a broadly neutralizing antibody. And without going into too much detail, we know through a lot of studies that these bNAb precursor B cells are very rare. They have specific genetic features that are shared with each class of bNAb. Each class has a different set of genetic features that lets it do its job to bind to the HIV envelope the way it binds. But for each bNAb class, there's also a diverse pool of bNAb precursors, because although they all share certain genetic properties, otherwise they're very diverse, and different people make different bNAb precursors. And finally, these bNAb precursor B cells are just difficult to activate with wild-type HIV proteins. So we need to come up with a strategy that starts with these red cells among all these blue cells and activates them and then drives them to turn into long-lived plasma cells. And the way and Glenda mentioned the AMP trial, and we know from the AMP trial, from that one data point, that to protect humans against infection by HIV in the real world, you need bNAbs. 90% protection in humans requires high concentration of bNAbs in the serum, corresponding to an ID80 titer of 200. That's a lot of antibody. It's a very high bar, but at least we know from a clinical study what bar we need to reach. So that's a good thing. Our strategy for inducing broadly neutralizing antibodies is called germline targeting vaccine design, and it involves a sequence of vaccinations, as Lindsey mentioned. It starts, very importantly, with the priming immunogen that is designed to engage those naive bNAb precursor B cells that are very rare and that have genetic features that allow them to turn eventually into a bNAb. But the job of that priming immunogen is simply to generate a population of germinal center and plasma B cells that have gained a little bit of mutation in the right direction and can be activated and further boosted in the right direction by a different immunogen. And we believe we're going to need a succession of immunogens to take each of these steps to generate a wave of germinal center reactions to go all the way to inducing long-lived plasma cells, as I mentioned. And I'm going to show you most data today about priming and a little bit of data about giving the first boost in a human clinical trial. So our lead, as I mentioned, we're working on all the different classes of broadly neutralizing antibodies, or 4 different classes, but our lead project to test germline targeting vaccine design is VRC01 class bNAbs against the CD4 binding site. These antibodies, we have structures of many of them. This is a depiction of one of them binding to the HIV spike in the same place that CD4 binds. They engage the GP120 CD4 binding site. In order to do that, in this specific binding mode, they use a specific human heavy chain gene, a VH12 heavy chain gene, and they have a very short CDRL3, light chain CDR3, 5 amino acids. With those 2 features, you can have many different sequence properties otherwise, and you can bind this CD4 binding site in this binding mode. They have diverse heavy chain CDR3s and light chains. So to get the process started of eliciting these bNAbs, we need a priming immunogen with appreciable affinity and avidity for diverse VRC01 class human naive precursors that have these two genetic features, but also have diverse heavy chain CDR3s and diverse light chains. And through lots of work starting back in 2010, we developed this, my colleagues and I developed this immunogen called EOD-GT860mer that is a self-assembling nanoparticle, as shown here, presenting 60 copies of a mini domain that has been engineered to have affinity for VRC01 class precursors. And this nanoparticle does have appreciable affinity and avidity for diverse precursors. It primes VRC01 class precursors in stringent mouse models, and it can induce memory B cells that can be further boosted toward BNAB development in mouse models. And we've published extensively on that. But after all of this work, the big question was, okay, you know, all this is great science, but if it doesn't work in humans, it doesn't lead us anywhere toward a vaccine. So along with what Glenda was saying. So we tested this immunogen as a protein adjuvanted with AS01 in a human clinical trial, IAVI G001, and we published the data in 2022. And that data provided proof of concept in humans for germline targeting priming. The molecule, the vaccine, induced VRC01 class precursors in 97% of vaccine recipients in this clinical trial. And I would hope it would be great if you could go check it out, read it. There's a lot of great data in that paper. I don't have time to review all of it. You can see we had a placebo group where only 2 of 12 placebo recipients produced VRC01 class responses, and but 35 out of 36 vaccine recipients did. And the only person that didn't, we found out they weren't genetically capable. They didn't have the right allele of the VH12 gene. And I just want to show a little bit of data there. So in these graphs, we're understanding how did the immunogen actually succeed, and did germline targeting priming strategy actually work? So in these graphs, this is from the low dose group and from the high dose group. And on the x-axis, each symbol is the total number of VRC01 class sequences we recovered from one vaccine recipient in the trial. So this person had around 150, 160 VRC01 class B cells that we isolated and documented. And on the y-axis, what we're showing is we've done bioinformatic analysis of those VRC01 class sequences, and we've clustered them such that if 2 sequences are in the same cluster, that means they came from the same precursor. They're very similar in their sequence features. They have very similar HCDR3s and very similar light chains, for example. And from this, we can see that the median number of so each number of clusters is like how many precursors, naive precursors, were actually triggered in that individual. And so we can see in the low dose group, the median number of precursors triggered was 32. And in the high dose group, it was 65. And so this data showed us right away that actually vaccination triggered many different VRC01 class precursors, started the process of BNAB elicitation. And we think that that result accords with the fundamental hypothesis that the immunogen needs to trigger diverse precursors. Each of these precursors were quite different from each other. And so that helps us understand why did it work, not just showing that it did work. And I want to give one other data slide from this trial, and just focus your attention on this panel of data right here. What I'm plotting here is we did induce VRC01 class responses, and we looked at the sequences of those B cell receptors, and we reverted all of the recognizable mutations back to what we believe was the germline precursor that was triggered by the first vaccination. And then we made those antibodies, and we measured their affinity for the EOD-GT8 immunogen. So on the y-axis, I'm plotting the dissociation constant, or the affinity of each of those precursor antibodies for the immunogen. And in the trial, when you vaccinate with an immunogen, yes, we got VRC01 class responses to the CD4 binding site, but we also got all kinds of other responses to the CD4 binding site that were not VRC01 class. In fact, VRC01 class responses were in the minority. We induced lots of them, but 75% roughly of the B cells that we got that were CD4 binding site specific were not VRC01 class. So we also looked at those sequences, reverted their mutations, and made those antibodies corresponding to those precursors. So what we're plotting here in blue are VRC01 class precursors from the trial and how tightly they bound to the immunogen. The median affinity was around 100 nanomolar. And the competitors, which were much more common, these are the green dots, and their median affinity was 100 micromolar. So you can see also from this data that the immunogen that we designed to have high affinity for the targeted precursors and better affinity for those than for the competitors, it did do that. It had much higher affinity for the VRC01 class precursors than for the competitors. And then we made VRC01 class and competitor responses at multiple time points after vaccination. In the trial, people were vaccinated at week 0 and week 8. And you can see that the blue data, the VRC01 class responses, has much better affinity, about 3 orders of magnitude, than the green data, which were the competitor responses. So this affinity gain, this affinity gap was maintained. And you can see that germline targeting design basically substantially altered the competitive landscape in favor of the targeted VRC01 class response. So I wanted to show those little pieces of data just to say, not only did the vaccine do its job that we want it to do and track with what happened in mouse models, but we can look at the data from the human responses and understand it worked for the reasons that we thought it should. And that's very helpful. That means other people can use the same strategy, and we can all keep working on germline targeting vaccine design to try to make an HIV vaccine. So I think these are very important findings. We also found, I mentioned that we're trying to induce binding with a particular binding mode, and we have lots of structures from Peter Kwong and Pamela Bjorkman and others about how VRC01 class antibodies bind to the CD4 binding site. In this clinical trial, working with Ian Wilson and colleagues, we solved crystal structures of multiple different VRC01 class responses elicited in the clinical trial, and the binding mode agrees exactly with what the mature BNAbs look like. So, and we knew that from the sequences anyway, but this provides formal proof, which was also very, very good. So IAVI-G001 establishes proof of principle for germline targeting vaccine priming in humans, but that's not a vaccine. That's not inducing BNAbs. That's just doing this one start. It's a priming. It's very effective. It's very consistent. It does high frequency. It induces a pool of responses that are at high frequency. It seems like you should be able to boost them, but we need to find that. We need to show that in humans. And I'll just remind you about this whole strategy. We said we think we need to give a succession of different immunogens in order to induce broadly neutralizing antibodies. That implies that we know that giving a heterologous boost, like we're depicting in this diagram, can be very effective and can be done very consistently and work in everybody that you vaccinate. But we don't know that, actually. No one has ever shown that in humans, that that would work in a germline targeting vaccination sense. So we think the next major milestones are to demonstrate that heterologous boosting toward BNAb actually works in humans. And I'll show you some data about one heterologous boost in this same pathway. And then the next one after that would be, we need to induce BNAbs, at least one class in humans. And then to make a vaccine, as we've said, we need to induce multiple classes in parallel. So you can imagine, as we mentioned at the beginning, trying to do all that and learn from human clinical trials is going to take multiple phase 1 clinical trials. And if you have to make GMP protein to do all those clinical trials, it's going to be expensive and it's going to slow you down. So we've been partnering, Scripps and IAVI have been partnering with Moderna for quite a long time to try to accelerate HIV vaccine development and clinical testing. And mRNA vaccine delivery allows you also to deliver envelope as a membrane-anchored envelope instead of a soluble one, just like the spike vaccine was. As the native trimer is presented on a virion, so we always thought, well, that might be an advantage, and I'll show you some data about that. So I'm going to show you now data from mRNA clinical trials. So the first question was, if you use Moderna mRNA with that same priming immunogen EOG-GT860mer, will it still prime VRC01 class responses as we showed in G001? And what this slide is showing you is, yes, it does. And it works both in the United States and in Africa. And we've in South Africa and Rwanda, and we've put a really high priority in showing that germline targeting works in African populations because that's where a vaccine is needed the most. And what we're showing here is on the x-axis is time after vaccination. Everybody was vaccinated at week 0 and week 8. And on the y-axis is the frequency of VRC01 class memory B cells among all IgG memory B cells in the blood of the participants, in the PBMCs. And you can see at week -5, before any vaccination, the frequency of VRC01 class B cells is really low, and many participants don't have any. But after one vaccination now, and what we're showing now in blue is the original data from G001, and in green is the data from G002, that was mRNA in the United States. And in red, in this pink color, is VRC01 class responses from African individuals in G003 induced by RNA. And you can see that RNA in both G002 and G003, actually with this dose of mRNA, the frequencies were even higher than they were in G002. And the percent response among responders was super high, 94%. And the only people that didn't produce a VRC01 class response in G002 and G003 were, again, those that were genetically incapable. They did not have a permissive VH1/2 allele. And so great, things work the same way with RNA. In fact, a little bit better. It's more immunogenic. And so then the question was, can we then test the next concept? Can we use the fact that mRNA manufacturing gets us back to the clinic more quickly? So in the same clinical trial, we were able to give a heterologous boost and see if that concept worked. I'm going to talk about that briefly. So in G002, as I mentioned in group 1, we gave 2 shots of EOD GT860 delivered by 100 micrograms of RNA, Moderna RNA. But the rest of the trial was aiming to find out if we give a heterologous boost, does that advance the B cell maturation toward BNAb development? So group 2 had 1 shot of the prime and then 1 shot of a heterologous boost, core GT2860mer. Group 3 had 2 shots of the prime followed by a shot of the heterologous boost, and we had reasons to believe we needed to try that. We thought that might be the only way it works. And group 4 was giving the heterologous boost only. And I'll tell you right now, the heterologous boost only doesn't really induce any appreciable frequency of VRC01 class responses. So I'm going to go through the immunological data first, and then I'm going to touch on the safety data and talk about what our next steps are after that. And I'm going to fast forward through lots of data. Both G002 and G003 were published very recently in May in Science in 2025, so I encourage you to go take a look. In G002, we tested these heterologous boosts. We found VRC01 class responses after the boost. We could tell by looking at the sequences that they had gained somatic hypermutation more than just after the prime. And we could see for VRC01, we know which mutations to look for, so we could identify a subset of the VRC01 class responses that had particularly high quality VRC01 class responses, and we called them elite VRC01 class responses. And we made sort of the regular VRC01 antibodies and the elite VRC01 antibodies, and we found that the elite VRC01 antibodies actually had appreciable neutralizing activity against pseudoviruses that are 1 mutation away from wild type. And that's what this table is showing. It's showing all of the elite VRC01 class antibodies from this group EOD, 1 shot of EOD followed by 1 shot of core. And it's showing that they don't neutralize wild type viruses at all, but they do have appreciable activity, neutralizing activity against these viruses that are mutated to remove the N276 glycan, which is a barrier to VRC01 class responses. And these are also highly sensitive VRC01 class. These viruses are highly sensitive to VRC01 class responses. Nevertheless, this was super encouraging. And this is clearly, you can appreciate, this is evidence that the vaccination, the heterologous boost, did move the B cell responses toward BNAb development. They're still not BNAbs, but they're kind of, teenager. They're developing in the right way. And if you give 2 shots of the prime, you don't get this behavior. And so I'm just going to give you one more slide about those responses. As I mentioned, we called them elite VRC01 class responses, and we can identify them by looking at their sequences because they had 5 or more what we call key VRC01 class heavy chain residues. Those are the ones that had the neutralizing activity. And so we decided, well, let's go, since we now see that these actually are neutralizing antibodies against, near native viruses, we want to know, well, how common were they in all of our sequence data? We couldn't make, we have thousands of these antibodies in terms of sequencing. We only made like 75 of them for neutralization testing. We couldn't afford to make thousands. But how common are they? And so one question is, well, if only a few participants are making them, this is not particularly meaningful. And so what I'm showing you here is for the different regimens in the clinical trial, and I'll call your attention to this 1 shot of EOD followed by 1 shot of core at week 16. So that's just week 0, week 8, now week 16. You can see I'm plotting on the y-axis what % of participants produced an elite VRC01 class response, and you can see it's more than 80%. And then that same regimen, EOD to core, if you just wait out to week 24, now it's more than 90% of the participants have an elite response. So that is good. That is something that we'd like to see for like a bona fide vaccine. You need to see consistent responses. Then the next question is, okay, you see these responses in 90+% of the participants, but how frequent are they? I mean, if these people are just making like 1 or 2 of these B cells, they would count as having made it, but it wouldn't be so promising for a next booster to find those B cells and send them further toward BNAb development. So what I'm plotting here is, again, on the x-axis, all the different regimens, and I'll call your attention again to 1 shot of the prime followed by 1 shot of the boost at week 16. And on the y-axis is the frequency of elite VRC01 class B cells among all memory B cells in the PBMCs of the participants. And you can see the median frequency is 1 in 28,000. That, I'll just tell you, is a high frequency. That is much higher than the precursor frequency in the first place of naive precursors, for example, and you would expect to be able to boost that. And you can see if we wait till week 24, it's even higher. It's better than 1 in 10,000. So yes, the elite precursors occur in almost everybody, and they're very common. And then the final question that we had was, okay, that's great, but if these elite precursors it's a small fraction of all VRC01 class responses, are we just talking about a single clone derived from 1 precursor, or are we talking about a highly polyclonal response? If it's in each person is only just derived from 1 precursor, that wouldn't be as good, because we want a very diverse VRC01 class response developing in these individuals. We want a polyclonal BNAb response so that HIV will have a harder time escaping. And so what I'm showing you here is, again, the different regimens. And now I'm showing you the % unique clone. So 100% means every single clone, elite VRC01 class clone, is unique and is derived from a different precursor. And you can see, again, in the EOD to core group at week 16, 85 or more % of all of these elite responses derive from a unique precursor. So we're getting these amazing VRC01 class responses that have ability to neutralize near-native sensitive viruses. They happen in almost every participant. They're very common in almost every participant, and they're derived from very many different precursors in every participant. I don't think we could have asked for more in terms of the immune responses in this clinical trial. So we're very excited about it. We also interrogated some of these antibodies structurally, and we showed that they bind to HIV trimers in the exact same way that VRC01 class BNAbs do, and so as you would expect from their sequence properties. And so the conclusions are, IAVI-G003 extends proof of principle for germline targeting priming to Africa, where an HIV vaccine is needed most. And IAVI-G002 provides clinical proof of principle for germline targeting heterologous boosting. And frankly, the practical responses are very encouraging, as I just showed you in G002. I just want to mention, I'm just going to really quick. We conducted one other clinical trial that's highly relevant to germline targeting vaccine design, and that's with the HVTN. And we said most of the boosts that we give in a germline targeting regimen are going to be HIV trimers. And so if you're going to use RNA to deliver trimers, should it be a soluble trimer or membrane anchored? And the trial showed it should be membrane anchored in this trial. The last quick finding that I need to mention is the safety finding. We can't skip it. I'm sorry, I'm taking a little bit longer. Is in G002 and HVTN302. The overall reacto and safety profiles were generally favorable, but some participants did experience urticaria. In G002, almost 12% of participants reported urticaria, and 10% it was chronic. And in HVTN-302, 6.5% reported urticaria. In G003, there was no urticaria, but 2 participants reported pruritus. And the details of the urticaria had been published in Willis et al. And in Riddler et al. And in another paper coming out by Parks. And so I will not go through those details any further. But for further development of these vaccines, we need to try to find conditions in which people are not getting, the rate of urticaria is much lower. We showed in Willis et al. That the rate of urticaria in G002 and HVTN-302 is vastly higher than any other Moderna vaccine. So I encourage you to check that out. I don't have time to go through it. And we also showed in the Willis et al. Paper, we have a lot of Moderna platform safety data. We also showed that the number of Moderna vaccinations doesn't lead to higher frequency of urticaria either. So there's something about these HIV antigens that mixed with RNA was giving urticaria, and we still are trying to work on with many colleagues what that was. One thing that we can try to do is in our next clinical trial, we're coming back with the same immunogens at a lower dose. And the question is, at a lower dose, can we get those same favorable immunogenicity responses, but much less urticaria? And I'll just mention that instead of the wild type trimer test in HVTN-302, we're testing a germline targeting trimer. And I will just thank all the people involved in G002, G003, obviously tons of people involved in both studies in planning them and executing them, and lots of sponsors and collaborating institutions. And same for G0 and HVTN-302, many people involved in the trial, in the protocol, and the site and the staff, and obviously thanks to the participants. And I'll just stop there. Sorry for going long.
No, thank you, and you can release your screen. We're running late, but I just want to ask one question from the audience, brief answer if possible, and then we can move to the next speaker and potentially have more questions at the end. One of the questions written in earlier made the point that given HIV as latency as a retrovirus, if a single virus infects a cell, then have you established infection? So is it possible from a vaccine standpoint to prevent that single event from happening?
Yeah, 2 parts to the answer. One is that in NHP studies, and there have been lots with Schief, which is like SIV with an HIV envelope, many different, Dennis Burton, John Mascola, many investigators have demonstrated that bNAbs, if present at sufficient concentration, can provide sterilizing immunity, meaning you cannot detect any viremia, you can't detect any cells that were infected. Are we going to achieve sterilizing immunity through bNAbs in humans? We don't know, quite possibly not, and that's what the question is about. And that is one reason why I mentioned that broad T-cell immunity is likely going to be necessary. If you can imagine, we put up a big wall with bNAbs and we knock down almost all the virus and a few squeak through and do infect cells, then we'd like to have a backup team, if you will, of broadly reactive T cells that will find and kill those HIV infected cells early enough before the infection can be disseminated. And, you know, whether or not that's possible is an active area of research.
Thank you. Terrific science, terrific explanation. Let me pass it back to Dr. Mgodi, and we can move to our next.
Thank you so much, Dr. Schief. And so our next speaker is Gilbert. Peter, I'm just trying to get to his, we've worked with Peter quite a while in the studies, and very soft-spoken, but highly efficient. So Dr. Gilbert, he's from the HIV, he's a professor of biostatistics at the Fred Hutch Cancer Center, and he works closely with the HVTN and also the University of Washington. He conducts statistical science research for randomized trials of candidate vaccines for HIV, SARS-CoV-2, and other infectious pathogens. He specializes in statistical methods for analyzing these trials in pursuit of understanding immune correlates of protection. And he's one of the team members who worked on the famous PT80 that stemmed from the AMP study. So well done, Peter. And that was epic. So he's a central application of his research is development of biomarker surrogate endpoints, as I've just said, that can be used to infer the level of vaccine efficacy against infectious diseases. Dr. Gilbert is a principal investigator at the Statistical Data Management Center, the SDMC, for the NIH NIAID-sponsored HIV Vaccine Trials Network. Over to you, Peter. Thank you.
Okay, thanks, Dr. Mgodi and Shan and others. Yes, I'm going to speak about correlates of immunity for HIV vaccines, focusing on V2 antibodies. And this is our team of collaborators on this project with Zoe Moody and especially with Susan Zola-Pasner and Georgia Tamaris. The data come from the Tamaris lab. I'll be looking at correlates across 4 different HIV vaccine efficacy trials that took place between 2003 and 2021. And there's so many people to thank for this work from the study participants and the Global CABs community members and the protocol study teams and the laboratory scientists. To frame this topic, I'm grateful to Bill and others for tirelessly pushing forward BNAB listing vaccines, because I think it's very likely that neutralizing antibodies will be a correlate protection for HIV exposition. And so it's great that the field is focused on that area of development. And I also think it's true that the V2 antibody correlative protection hypothesis remains alive. Previous studies have looked at V2 antibodies correlates in the 4 individual FC trials I've mentioned. And over the last handful of years, there's been new causal inference statistical methods that have been developed that can provide new insights into COP, as well as new structure prediction tools. And so what I'm going to do today is show how this group has been applying these technologies to the 4 FC trials to look for additional insights about V2 antibody correlative protection. And the overarching question I'm getting at here is, someday can we have a vaccine that is looking at, well, inducing V2 antibody effector functions as well as BNAbs and T cells to get a really good vaccine? And Glenda noted for us that the ARVN-4 trial showed efficacy, modest. It was 26% in the per protocol cohort. Also, the 7-5 vaccine regimen trended toward positive efficacy. And then there wasn't evidence of efficacy in the 505 and the 702 trials. And this is reminding us that there has been V2 antibody correlates papers published for the HV10 FC trials, as well as a suite of papers really for the ARVN-44 trial. And today we're focusing on the IgG and the IgG3 antibody measured from the Tamaris lab. And I'll first look at the peak immunogenicity. And the whole point of this talk is to get a head-to-head consistent analysis across the 4 trials. And so we're restricting to the 4 V2 antigen reagents for which we were able to obtain identical data from the same assay based on a case control sample from each of these 4 studies. The 4 antigens, there's the case A2 conformational subtype B antigen that Dr. Zola-Pasner selected as the primary antigen for the ARVN-44 study. And then the other 3 antigens were vaccine inserts for the ARVN-44 vaccine or for the 702 vaccine. So we have the antigens representing clade B, AE, and clade C. And looking at immunogenicity, so this is for case A2, for total IgG, response frequencies on top, magnitudes on the bottom. So V2 antibody were low for the 505 regimen. They were modest for the 702 regimen. They were pretty good for the 705 regimen, and they were really high for the ARVN-44 regimen. And also recall that the ARVN-4 regimen was also tested in a South African population in the 097 study. And interestingly, the V2 antibody responses tended to be higher in the South African population than the Thai population. These are the same data except for the AE, A244 antigen. And interestingly, the 702 regimen now has considerably higher responses than against case A2. The 7-5 responses are about the same. The ARVN-4 responses are always the best across all these antigens. And we see this also for the subtype C antigen. And this is particularly interesting because, the 702 and 705 vaccines, they had a clade C boost to match the circulating strains. And the R404 vaccine did not have a clade C insert. But despite that, the R404 vaccine still had the highest, cross-reactive clade C responses. And now I'm looking at IgG3, and see the response rates were pretty modest for the KSA2 antigen. Most people didn't have a IgG3 response. But you do see in 705 and R404, there's these rare subgroup vaccinees that did have responses. And we see that for the A244 antigen, we start seeing many more responses, especially for the R4 regimen. And for subtype C, again, you see pretty good responses and diversity of responses for R404 and some as well for 705. And so in conclusions for immunogenicity, the R4 regimen has got the greatest IgG and IgG3 responses across the clades compared to all the other vaccines. However, there was quite a bit of diversity in responses for the 705 regimen with a minority subgroup with high and cross-reactive IgG3 responses. So that means we can study a correlate pretty well in 705. For 505, basically, we can't even study V2 antibodies as a correlate. The responses are simply too low. For 702, the responses are modest, but there's enough there that we can possibly find a correlate. So now I'm going to move toward the next section. Let's look at these V2 antibodies correlates of protection consistently across the 4 trials. So the first technique I'll use to study a correlate of protection is the classic trial level or meta-analysis surrogate endpoint evaluation technique. It's a very well accepted approach to evaluating a surrogate endpoint across biomedicine. In general, this approach studies the association of treatment effects on an endpoint of interest and on the putative surrogate endpoint. So one classic example applied to HIV therapeutic trials, it's been used to validate that HIV viral load is a surrogate endpoint for progression to AIDS and death. And here I'll be doing the analysis by looking at the association of vaccine efficacy with the peak geometric mean V2 antibody in the vaccine arm. So here's the results. The top panel is the results for the IgG against each of the 4 antigens, and the bottom panel for IgG3. So we're looking at vaccine efficacies here as a function of the geometric mean V2 antibody. And so the picture here is pretty simple to interpret. Basically, there's an increasing pattern that vaccines that had higher geometric mean V2 antibody, they also had higher vaccine efficacy, and that's consistent across the different antigens. So this is positive for V2 antibody surrogate endpoint. And now I'm going to move toward individual level causal inference techniques for studying correlate protection. The first technique is called controlled vaccine efficacy, where we estimate at a given antibody value S. We're asking the causal question, what is vaccine efficacy if we assign all participants to get vaccine and to have the antibody fixed at a certain level S versus assigning all participants to placebo? And then the second causal inference approach, we're asking the question, what is vaccine efficacy if we assign all participants to vaccine and to antibody thresholded above a given value compared to assigning all participants to placebo? And these methods, they've been applied to COVID studies and dengue and influenza over the last few years. And now we're getting around to applying it to these HIV vaccine efficacy trials. So this is showing the results of the controlled VE for the 705 and the R4 trials. There's 2 curves on each plot because one is a smooth curve estimated via Cox modeling. The other is an unparametric curve, which allows the shape to flexibly change. So it has the ability to detect thresholds. And the result for 705 is that there's some increase in vaccine efficacy with the V2 antibody. It maxes out at about 35% efficacy at the highest V2 antibody levels. And for R144, there's a more dramatic change where there's a significant increase in vaccine efficacy with V2 antibody. And for those with the high antibody levels, the efficacy reaches about 70–80%. Now, this is showing the results thresholding by the antibody above a threshold. So in these plots, the blue line is the vaccine efficacy for the thresholded groups. And then the green line is the reverse cumulative distribution function, which shows us what percentage of vaccinees have the antibody value above the level listed on the x-axis. And this plot is for the KSA2 antigen. And basically what you can see is that there is some increase in vaccine efficacy for both trials with increasing the threshold. So again, it's some evidence of a correlate. And this is focusing on the ARB-144 trial, looking at all 4 antigens. And we do see some increase in vaccine efficacy with the level, with the thresholding for all 4 antigens. And then we also pooled the data between 705 and ARB-144. This plot is looking at IgG on the top, IgG3 on the bottom, and it's looking at the 2 antigens, KSA2 and A244. And again, we see that for all the analyses, vaccine efficacy increases with the threshold. And in fact, for all 4 of these panels, the VE curves increase to values that are above 50% vaccine efficacy at some threshold. So this is all encouraging toward the V2 antibody correlate. And now, with Bill on the call, I have to definitely highlight the word preliminary, that our group is learning structural biology to predict protein structure from sequence, because we think this can improve our insights into correlates of protection. And so I'm going to show some preliminary investigations of basically using deep neural networks to predict structure from sequence and use that to get insights into correlates. So we're using AlphaFold 3. We can use this to assign structural features to HIV amino acid sequences. One feature we're looking at is a property of an amino acid position. It's its depth. What we do is we input an HIV amino acid sequence, calculate its accessible surface area. We roll a probe of radius 30 angstroms, which represents a potential antibody along the surface of this protein. And then the depth of a position is basically what's the distance of the closest probe. And what we're going to do is look at the accessibility of the immunogens in these 4 FC trials to potential antibody by calculating the depth at every amino acid position across the sites 120 to 24, which is the V1, V2 region. And here's the results. Hard to read, but there's 14 panels here for the 14 different immunogens in these studies. But there's 2 points I want to make. One is you can see that these lines undulate up and down a lot. That's just saying that some positions are much more accessible to antibodies than others. Some are relatively buried, some are right on the surface. Then another thing we can surmise here, if we simplify this and look at the average depth of positions, and we look at those across all the immunogens grouped by the efficacy trial, we actually don't see appreciable difference in surface accessibility across the immunogens. Because one of our questions was, would we find that the ARB-144 immunogens were more accessible than the other images because the efficacy was higher, and we didn't really see that. However, for those in the field for a long time, we know that position 169 in V2 is somewhat famous because there were Civ studies and correlate studies and mechanistic studies from Duke that have showed that likely ADCC activity against this epitope had to do with the ARB-144 protection, and it depended on having a lysine at site 169. And I'm just pointing out here that based on AlphaFold 3, site 169 is one of the most surface exposed sites on the V2 protein. And then the other thing we did with AlphaFold 3, we used template modeling scores to compare immunogen predicted structures to the predicted structures of all the placebo sequences that were endpoints in these trials, because those placebo sequences represent the circulating strains during the trial. And basically, we're looking to see if some of the immunogens had better structural match to the circulating strains in their trial compared to other trials. So here's the results where, we have a box plot of TM scores against the circulating strains for every single immunogen. So you can see there's variability in these structural similarity levels across immunogens. And to summarize some points, for ARB-144, what we see is that the AE prime 9-HT-TH33 and the AE boost CM244, they have a high structural match to their circulating strains, whereas the B strains did not have high match. Not surprising given that AE was the dominant circulating strain. We also see that for 705, the boost C97A had pretty good structural match, but neither the mosaic primes had good structural match. And for 702, we see the boost 1086 had good structural match, but the TV1C boost did not, nor the ZM96 prime. And then I want to highlight that when we redo these structural predictions by taking out the hypervariable loops, the TM scores get much higher. So this is showing us that a large amount of the structural mismatch is due to the hypervariable loops. Because in reality, these loops are flopping around, and the structure predictions we have right now are for a static structure. So, and also I think this picture here, it's highlighting that, you know, it's only for the R144 regimen that we get a good structure match for both the prime and the boost. And I think Shan Liu's talk might say something about the importance of that kind of match. Okay, so to conclude on our AlphaFold work here, we're finding that the 4 vaccine regimens do not have clear differences in their surface accessibility to potential V1 and V2 antibodies in terms of depth. We're finding in terms of these TM score structural similarity measures that only R144 had a good structural match for both the prime and the boost. And we're wondering if this is a clue of one of the reasons why the R144 regimen may have had the highest efficacy. And for 705, we learned that the mosaic prime inserts didn't have that great structural match. And remember, these mosaics were optimized for their 9-mer linear peptide match, and that's a different thing from a structural match. And then I mentioned that we're getting static structure, so we really need to move on to look at molecular dynamics, and that's the next project. So now, lastly, I want to say something about where are we headed? I mean, if someone can develop a vaccine that gets better V2 antibodies, and Shan is going to talk about one in a moment, but if we can do even better than what Shan is going to show us, can we get to a, you know, pretty good efficacious vaccine based on V2 antibody? And to address this question, it's a specialty of our group to think about data fusion and transportability and to try to predict vaccine efficacy based on a surrogate endpoint. So recently we published a paper on a statistical method for doing this kind of prediction. It does data fusion across 2 data sources. Data source 1 is from an HIV vaccine efficacy trial, from which we learned the relationship between a surrogate and the risk of HIV. So any of the efficacy trials I've discussed is an example of data source 1. And then data source 2 would be a surrogate immunogenicity trial where we get the immunological marker measured for both an investigational vaccine and the source vaccine that was studied previously in the efficacy trial. And then we fuse the data together to estimate a relative vaccine efficacy of the investigational versus source vaccine. And I'll just say that the way this works is we have to do transport. We learn the relationship between V2 antibody and risk in the efficacy trial, and then we transport that, assume that same relationship holds in the immunogenicity trial. And then we do a second transport where we assume that the relationship between the surrogate and outcome in the source vaccine also transports to the investigational vaccine. And under those 2 transport assumptions, then we can predict the relative vaccine efficacy of the investigational versus control vaccine. And our key assumption is really that the surrogate works, that basically after you control for V2 antibody, that whether you're getting the investigational or the source vaccine doesn't matter for risk, that the antibody level is what determines risk. I'll skip these for time. And I want to point out that Moderna actually just very recently published a paper on its flu vaccine, which did support this surrogacy assumption that we need for our method here. And you can see that for looking at the risk of influenza-like illness as a function of HAI titer, that the curve is basically the same for the inactivated vaccine and for the mRNA vaccine. And this is showing that the surrogacy assumption was supported by the data. So now I'm going to apply our method to the H124 trial, which I'm quite sure Shan will talk about because it's a vaccine developed from his group. And I'm going to ask the question, what is relative vaccine efficacy of this DNA protein vaccine versus the 705 vaccine regimen based on this data fusion method? And so the Tamaris lab, they ran V2 antibodies in samples from these 2 trials using a common set of antigens. I'm going to focus on the IgG3 Brett score, which is a weighted average of readouts to these 5 antigens. And so the immunogenicity data, basically the responses are considerably higher for the 124 vaccine regimen compared to the 705 vaccine regimen. And here's the results of our application of our bridging method. So basically, had the 124 regimen been studied in 705, our answer is that the relative vaccine efficacy would be 21%. So 21% better than the 705 vaccine. And then we can ask questions like, if the 124 vaccine could increase the V2 antibody level by a log or 2 logs, we can estimate that the relative VE would have been 50% or 63%. And if we translate this to absolute VE versus the placebo arm in 705, we can say that the 124 regimen would be predicted to have 29% absolute VE. And if you can increase the response by a log or two, we can get up to 58 to 69% efficacy. So the conclusions of this talk, total IgG and ID3 antibody to V2 antigens are showing evidence as COP using multiple approaches. We think it's appropriate to state that the V2 loop, is a vulnerable antibody target for HIV vaccines, definitely for BNAS, but also for FC effector function antibodies. And the surrogate-based modeling is predicting that if vaccine research supplies improve V2 antibody levels, then we can start to believe that it's rational to think we could get efficacy in a meaningful range. Okay, and thanks again to an enormous set of contributors to all these FC trials over the years.
Thank you so much, Dr. Gilbert. A very interesting presentation. To start, and thank you for getting us back on track in terms of time. To start off the Q&A session, Dr. Gilbert, I'm going to ask you a very difficult question. Could you summarize your presentation for lay people, because I was watching the participants list, and I realized there are a number of community people, and in the HVTN we make a summary for the community, because I want everyone who's attending this symposium to have something to take away with them. So please start there, and then we'll come to the questions in the chat box. Thank you, Dr. Gilbert.
Okay, thank you. So point one, I would say that there's pretty good knowledge that if we get a vaccine that gets high levels of broad neutralizing antibodies, that it should work. Because there's many precedents of vaccines for other pathogens that get high and broad neutralizing antibodies, and they did work, and neutralizing antibodies are accepted as a core protection for approving vaccines. So that's very positive. What I talked about today was kind of an alternative correlative protection based on measuring antibodies to the V2 part of the virus, and these antibodies are not neutralizing. So it's kind of an innovative or avant-garde area of research. No one's really validated that you can get a accepted correlative protection that's not neutralizing. And then I noted that historically there's been a lot of effort put in by the Tomorrow's Lab and the HU10 and the ARBA44 TIE team to look at V2 antibodies as a correlate. And I've basically been saying that there is good evidence that, across these FC trials, that for the vaccinees that had high V2 antibodies, they had some protection from the vaccination. We can't prove that this is a correlative protection, but we have these promising leads that it's a correlative protection. So to me, then the take-home point is that we don't need to kind of sunset the entire approach to vaccines to try to develop V2 antibody responses that may or may not have neutralizing, potential. It's still valuable to try to get vaccine regimens that get higher and broader V2 antibodies, because in the future they might contribute to an efficacious vaccine. Because I think in the end I think we probably all believe that the ideal vaccine would have a combination of functions. It's going to have broad neutralizing antibodies. It's going to have Bill mentioned that T cells are essential, both CD4 and CD8 probably, and then also V2 antibody effector antibodies could be a bonus to improve the efficacy of the vaccine.
So Peter, along those lines, just looking at several of the questions in the Q&A, how do you know which antibodies to model, which antibody responses to model around? Because so many different kinds of antibodies get induced by a vaccine. How do you choose these to base your model?
Well, it goes back to, it goes back to some more comprehensive analyses that we're looking at V3 antibodies and looking at different types of assays and discovering that the V2 antibodies had the strongest prediction of whether someone acquired HIV. So it was empirical, discovery research. That's part of the answer. But I think the other part of the answer is we need to understand mechanisms. You know, I think we'll need to kind of combine this with some kind of animal study that kind of proves that changing V2 antibody levels or deleting them or something corresponds to loss of efficacy, or, you know, so we need to kind of combine these mechanistic toggle experiments with the empirical studies in these population studies where we can just discover what are the best predictors of HIV.
Good. Thank you so much, Dr. Gilbert. May I also encourage you to continue perusing the Q&A and answering as many questions as possible. Dr. Baden, may you please introduce our last presenter, last but not least. Thank you.
Thank you, Dr. Mgodi, and thank you, Peter, for a terrific exploration of correlates of protection. Our next and final speaker, we all know well, Dr. Shan Liu. He's a professor emeritus in the Department of Medicine at the University of Massachusetts here in the U.S. He is a pioneer in nucleic acid vaccine development and has continuously worked to advance this field over the past 30 years. He continues to develop a heterologous prime boost vaccination concept and has developed a polyvalent DNA prime protein boost HIV vaccine, which in human clinical studies has demonstrated broadly cross-reactive immune responses. Besides being a U.S. boarded internal medicine physician for more than 20 years with clinical practice, he has managed major NIH-funded vaccine development programs, including GMP manufacturing, FDA IND filing, as well as execution of multiple clinical trials. He has broad biotech industry collaborations and joined the Worcester HIV vaccine group in 2022, serving as its chief scientific officer. He was past president of the International Society for Vaccines and is current co-chair of the Corporate Partnership Program at IAS. Shan, thank you for sharing with us another approach to elicit potent HIV immune responses.
Well, first of all, thank the conference organizer for this wonderful short but a very potentially important mini-symposium. This will be the first collaboration between IAS and ISV, and I'm honored to be a speaker today to talk about polyvalent DNA prime and protein boost HIV vaccine. In the next 20 plus minutes so, give everyone an update and share what we plan to do next. What did we start? Just 1 second. What did we start not from any fancy scientific hypothesis, because the vaccine actually after 200 years still is an empirical science. Many licensed vaccines, we don't even know their mechanism, never mind the epitope, that kind of questions. So what I started HIV vaccine many years ago, still going back to the fundamentals of a vaccine. That is, can you generate immune response that is strong enough? And the second question, can you cover the pathogen or variation of a pathogen a vaccine try to cover? I think those are the fundamental vaccinology questions. Frequently nowadays, we don't pay enough attention. In particular, for a challenging HIV virus as a target, we know for whatever the reason, generating antibody in a mouse model against a simple HIV envelope will not be so easy. Well, you can easily generate antibody against influenza, hepatitis B. So we need to have some new approach. What we use is DNA prime and protein boost. I will explain next. The second one, we all know HIV is a highly mutated retrovirus because they don't have the RNA for reading enzyme. So the virus mutated all the time. So we went back to the old vaccinology concept that's use a polyvalent antigen formulation, but we use a slightly modern way to develop such a formulation. Let's first talk about heterologous prime boost. The concept, this term is widely used now. I was the one 15 or 16 years ago, noticed from my own hands-on experiment, not just HIV, from many other studies in my laboratory, I found that if you try to deliver same antigen, but use different modality of vaccine, recombinant protein, DNA, viral vector, inactivated, killed, but you find that they always work better than just using one type. Then I wrote a review and also collect information people just did accidentally in history, in different studies, not just HIV. I found that this is a common pattern. Of course, after COVID, many people use this concept and this name become very popular. However, after careful search about all the possible options for HIV prime boost, we decided to use DNA prime and protein. One thing I want to emphasize, which I will mention at the end, we use a matched antigen, which is very important from B cell development point of view. The reason we selected DNA as a prime, because DNA as a prime vector, it has nothing else in the vector. It's a single HIV antigen or multiple antigen, but still it's HIV, so it's immune focusing. And also the antigen expressed in vivo, it will have the best chance for optimal antigen conformation. While recombinant protein, you have to go through denature, renature process, and very hard to make it original conformation as with the virus. And also our group had the first discovery. The DNA prime or nucleic acid vaccine to some degree have powerful potential in stimulating antigen specific development in germinal center, which people did not anticipate. The reason for that is we realized DNA vaccine actually induces both B cell and T cell. This T cell, especially TFH cell function, can stimulate the B cell development. When you give DNA vaccine, deliver both T and B in a one construct, it can induce the best antigen specific B cell or memory B cell. However, the DNA vaccine express amount of antigen is very low. That's why it doesn't stimulate a high level antibody response, although we have a lot B cell circulating. So what we noticed that if you give a protein vaccine, and that will stimulate many active B cell by DNA, then induce a very high level antibody response. We believe this is a perfect match. Both are very simple without other involvement. In our approach to DNA, we don't use a lipid nanoparticle. We use gene gun in early day, now we don't. We just use a needle injection. This greatly increased the safety profile as well as the manufacturing and the cost. So here's an example. You can see how powerful when we give DNA prime, your antigen specific B cell is like exploding. This is an antigen specific B cell spot in a mouse model. You can see antigen specific antibody secreting cell or memory B cell, they both in bone marrow spring. It's just everywhere. And this not just in mouse, in human trial, we use the same approach to collect a human PBMC and look that after enough DNA prime by day 100 or 150 is a huge amount antigen specific B cell memory. If we plot a curve, you think this is a very high level antibody specific B cell development and lasts very long. So the second question is, how do we develop polyvalent vaccine formulation in a modern day time? Older day, you just simply mixing different serotype antigen, and this has become very difficult for HIV because HIV has many subtype, genotype, and also we don't know which one representing the immune serotype. There's no consistent concept on that. So the question is, how do we establish antigen specific B cell memory? As Bill just talked about, actually the beauty of B cell development is a linear process. It's not a one step. So B cell can, from the early early B cell before a prematured or early B cell precursor to antibody affinity maturation goes through many steps. So this will be the time for us to generate huge diversity. If we mix multiple antigens, generate different B cells, especially with similar antigens, we know the issue is called the antigenic sink. That's why flu vaccine and COVID vaccine, we learn they are not easy to be boosted. Rather, they become very narrow focus. So if we have a multiple immunogen, we can start from the beginning, generate diversity of B cell population, and we can continue that trend getting broader and broader diversity. That's what we did. We studied in a rabbit model screening 80 HIV envelope antigen, did the pre-priming, and then with the boost protein and see their cross-reactivity. We found that not all the HIV envelopes have the same ability, general cross-reactivity. Only about 15% or 10 or 15%. In red color, they can cross different subtype. Another 20% may be narrowly cross-reactive, and then almost half of them do not cross at all, as in the white color. So what we did is select one from each subtype virus, clade C, A, B, and AE, and formulate a formulation. Here we did a bioinformatics analysis. Now maybe we can call the human AI analysis. You can see from the top, if we only have one antigen from each subtype, but against the 16 genotype, the match is very narrow, only when they come from the same genotype. However, if we just randomly mix any two antigens together, immediately the homology, as shown by warm color, will increase. If we have three or ultimately use 4 antigens together, you see they can cross-react or prevent any mutation of HIV. This is actually a simple explanation, because HIV, no matter how much mutate, they have a limit. HIV cannot mutate into influenza, right? So if we carefully select diverse enough sequence and mix them together, and they can predict or prevent any future automatic mutation, and then we don't have to worry this vaccine can be used today or future, or can be used in South Africa or used in China. So with this two progress in hand, we started multiple HIV vaccine program about 2 decades ago, more than 2 decades, funded by President Clinton's the program for HIV vaccine development, funded by NIH HVDDT. We did the first generation trial called the DPS-01. That was the first time we showed the DNA and protein combination polyvalent can induce a very bold immune response. And then funded by 2 more NIH funding, HIF-RED and IPCAVD. We move on to have the very important landmark HVTN124 study, as mentioned by Peter earlier. And then we published this data last year. And also we did the follow-up study with Lindsey's group at the Brigham Women's Hospital. Here, I will highlight what we found. This is the HVTN124 study. We use 4 envelope DNA plus a GAG. As the people mentioned, we want to have a backup T-cell immunogen, even though that's not our main focus. We use a protein drug product, our full GP120 protein matching the DNA sequence 100%. Use GRASE as a bedside adjuvant, and then deliver them by needle injection. So we will focus on the 2 groups in our Part B trial. T2, the blue color is DNA prime and protein boost. The red color is DNA protein delivered same time. First, I will show you the binding antibody, because this has been a major challenge in HIV vaccine field. Many trials, the titer, just the binding is low. Here, we can generate binding titer 10 to the 4 to 10 to the 6, and almost everyone, 100%, especially in the prime booster group. And this binding titer against any envelope we select or HVTN have the access. And then we next look at the ADCC as shown by RV144. The ADCC antibody could be another important parameter for protection. Here we use the infectious molecule clone, infect the cell as a target for the ADCC, which is very difficult in previous trial. And also we can compare the previous trial, their ADCC level versus the one in our study. You can see the prime boost was more effective, induce a potent ADCC antibody response. And then I just want to make sure people don't think we're not also working on neutralizing antibody. Actually, the prime boost, even with GP120, because they are expressing the antigen in vivo, they are highly conformational relevant. We can generate very broadly cross-reactive antibody. Here we use tier 1B viruses. Tier 1B virus is not easy to neutralize. You can see very broad cross-reactive. We do have one volunteer with a tier 2 virus, which we are not making a major statement here. And very exciting, we find out we can generate broad antibody against the V1, V2 antibody, as Peter just mentioned, as we learned from RV144. This could be a potential correlate protection. And the most important thing is the IgG3. For many previous HIV trial, IgG3 GP120, 70 V1, V2 is low. And this part actually is important for the immune correlated analysis. We also have induced a very high-level CD4 T-cell response, 100% volunteer in the prime boost group, and also they are highly polyfunctional. I want to highlight here the peptide we use in this assay not matching with our antigen. Rather, it's random selected global pool. And you can see because we use the full polyvalent approach, we can easily match them and induce very broad and high-level response. And the one thing very important, while everyone is trying to use old method induce neutralizing antibody against CD4 binding site, we were able to induce by GP120 monomer and DNA prime boost. Peter Kwong collaborated with us to confirm this is indeed a CD4 binding site antibody, and we could neutralize 10% of the 200 OA virus panel from NIH-VRC, including several tier 1B and several tier 2 viruses. This will be the first time any human monoclonal antibody isolated by vaccination, and that can neutralize tier 2 viruses without the glycosylation mutation. They are intact original virus, and they are also targeting at the most wanted CD4 binding site. And here is the crystal structure. They are not binding the same as the other well-known monoclonal antibody, but nevertheless, they can bind the trimer in a unique way to achieve this very important function. We also look at the many monoclonal antibodies we isolate from a volunteer. They have a potent ADCC activity. In particular, we try to see whether we can have anything like A32, which is isolated from HIV infected people. This is called the king of ADCC because it is highly potent. Here is the human monoclonal antibody 78. We isolate from one of a volunteer. It is a potent ADCC activity, actually can compete out against the epitope of A32. And amazingly, it shared the same the germline with A32. In other words, we generate a potent ADCC antibody, just like a HIV virus infection. They share the same structure, except the sequences are not identical. And the last piece data I want to share with you was a paper published about 2 months ago, led by Zoe Moody and others from HVTN. They compared the the immune correlate analysis among 13 HIV vaccine trials conducted by HVTN in the last 2 decades. I would just highlight one data here. You can see the far left, this is a 124-hour vaccine. The V1, V2 antibody score is over 1,000, but all the others are very, very low, including RV144, 705, and 702. 705 and 144 is a little bit better than the others. Maybe that's why they give Peter the chance to look at the immune correlate. I think that there's a confusion in the field. People say the V1, V2 series is dead because the 705, 702 had a good V1, V2, but did not induce protection. From this data, you can see the reason those trials failed or not induce high protection is they did not have V1, V2. So that's why this hypothesis, I think, is still valid and needed to be tested, especially with a vaccine that can induce very high V1, V2 score. Then we can decide whether the theory developed by Peter and his colleagues are truly a very important parameter predicting HIV vaccine. Also, I want to just show one piece of data, that is that antibody induced by our approach is a long duration and not drop that much. We first did this 10 years ago. We monitored 6 months, no dropping. And also the latest data we have not published with Lindsey's group at the Brigham woman, we monitored 12 months. You can see the antibody against each of the antigens are still high level, not dropping that much. So the summary of our vaccine approach, this vaccine, polyvalent DNA protein HIV vaccine, have a safety profile very high. I did not have a chance to show you all the details. The key part I want to emphasize, this is a highly polyfunctional antibody with binding antibody, V1, V2 antibody, ADCC antibody, and neutralizing antibody. By one vaccine, we achieve all 4 binding or 4 antibody objectives. At the same time, we maintain 100% CD4 T cell response rate plus diversity. And also antibody response is a long-lasting. Most important, I want to highlight the protection correlate. As Peter mentioned, IgG3, V1, V2, and CD4, the combination. And also multiple antibody isolated from this volunteer further support the validity or depth of this vaccine. So why people ask, I would just mention a little bit. Now we have the data, empirical data. We need to ask a scientific question. What happened? Why this vaccine is the most immunogenic and cross-reactive? I draw several, hypotheses here. The first, I think everyone so far in HIV field use a modified antigen. Believe it or not, nobody used the original HIV amino sequences. We are the only one using natural without single amino acid mutation. Maybe that's something we should cover as well. The second part, many people say the prime boost, we know a lot, but there's no difference. But that's not true. When you look at that, many of the prime boosts, especially people use the protein boost, because not all the proteins vaccine available readily, people use whatever they can get. So the match between prime and boost is very low. We analyze most of them are around 70. However, in our case, HVTN 124, we try to 100% match. You think about that. If a B cell being activated, you use a different antigen to stimulate, would that respond very well? So I think that this match is very important. And finally, I know people were disappointed with the Mosaic. That's such a wonderful design. Here, I want to show the difference between Mosaic and our approach. Mosaic design, as most, they have 2 antigens, but each of them is collecting pieces from different viral isolate, but mainly they still close to one clade B or C, but they disrupt the whole envelope structure. So the concern is may not be good for antibody because antibody depend on antigen conformation. This design may be good for T cell. Also in Mosaic, there's only one protein as a boost, 705. 706, they use 2. However, in our case, we use 4 pure, authentic, different subtype of antibody really generate true diversity and then further boost that. We think the antibody generated by this kind of prime boost will be millions in different subtype of antibody specialty. And finally, I think produce a boost with a high quality protein is very important. This is just example of our protein release test. You can see 100% pure, and also we have data showing the protein is stable for 7 years. With this, we hope we'll move our phase 2A study in South Africa. We hope to collaborate with Glenda and her colleagues, and also we'll continue a site at a Brigham Women with Lindsey's group. So this is my summary. I think outlook for an effective HIV vaccine, as we all learned from today, maybe we needed to induce diverse type of protective immune response, not just focus on one. Polyfunctional antibody activity may be important. We need a broad reactive against diverse HIV isolate, not just neutralizing, but the ADCC and the other antibodies also need a diverse. And finally, we can beyond the multiple one epitope and the long-lasting and the ultimate efficacy trial can answer our scientific hypothesis. This is work by many people. I will just quickly go through that, and people can find it from the recording. I will stop here. Thank you, everyone.
Thank you, Shan, for presenting another path to develop broad immune responses against HIV. A question alluded to by some audience Q&A, and I'd be curious in your response. Your approach is a tetrapentavalent natural sequence. How have you decided or determined that this is likely to elicit coverage of the incredible global diversity of HIV? How do you come to that determination that these sequences will get you to where you want?
Well, initially, this started from basic research laboratory, as I showed here. We just randomly selected 60, 80 envelope. It's a 10-year project. Many postdoc graduates in my lab, they have to take a part of this. This will take 10 years to finish. After that, we select A, B, C, E, one of them from the red color. Suppose they can cross-reactive. And then we use a sequence analysis against any of the top 16 HIV envelope consent sequence. We say if we use this full envelope, like the box here, against the 16 genotype, can we cover that or not? That's the mathematical analysis. That means HIV, no matter how they mutate, if they mutate too much within one subtype, they will be somewhat like other subtype, the basic. So in a human way, we did the AI, computer probably predict that. We did not use a computer. We used the human work and based on bioinformatics analysis. But then this is, of course, it's only theoretical. So what I showed you, the human data, is the proof. So far, you give me any envelope from anywhere in the world. I will show you whether I can bind or not, whether I can have an ADCC recognizing or CD4 T cell cross-reactive. That's what I can show.
Thank you. Another question, Shan. You've shown diverse immune responses can be elicited by vaccination. What advice would you give to Dr. Gilbert as to which of these immune responses should be modeled to create protection models? And how would you determine that?
Well, I don't think I'm qualified to tell Peter about what he should do. Actually, I was inspired and supporting me getting up every day in the morning to move forward in this challenging field. Is really his finding, his group's analysis on this one. showing we have some IG3, V1, V2 antibody, we can move forward. I think what he showed today, somewhat new, some data also very important. What he suggests with our current HVTN124 data, based on his model, he can predict we can have about 25 to 29% protection, which is not too far from RV144. He has not considered the neutralizing antibody. He has not considered ADCC antibody. If I add all those two parameters, maybe easily I can add another 25% protection. We will achieve a 50% minimum HIV vaccine, as Dr. Glenda Gray mentioned. So that's what I think we can do. Also, I am working with Peter closely. What I think we can do is to expand the V1, V2 antigen, not just the one so far used in his modeling. Unfortunately, it's a technical challenge, believe it or not. We don't have enough GP70 V1, V2 diversity in stock by HVTN. So my lab is producing a lot right now. So hopefully we can go back to do more analysis of the old efficacy trial, to do more objective, more comprehensive V1, V2 antibody analysis. The other thing I think I want to suggest, people asking, what is the really V1, V2 antibody tell us? Because we know they are not neutralizing, but actually we don't know whether that is neutralized because no antibody being isolated. So my group is trying to isolate some of V1, V2 antibodies since we have high titer. Also, I want to do a correlate analysis. Maybe V1, V2 antibody is not functional. It's just a surrogate marker, maybe just showing the antibody level is high, then you will see this antibody, especially IgG3. IgG3 is only 1/1000th of IgG1 in our body. We usually don't see them. Unless you have very good potent immunogen, you're not going to see IgG3 antibody. Maybe that's just tell us if your vaccine is strong, your antibody is high, give you a chance to see IgG3, V1, V2, maybe you will have protection. So it may be complicated, may be simple, we don't know. But this open the scientific field. Unfortunately, I hope NIH or other people will support people like us to move forward to ask such an important question for HIV and for general vaccinology.
Thank you. Dr. Mgodi, shall we wrap up the symposium?
Thank you so much, Dr. Shan Lu, and thank you, Dr. Gilbert, Dr. Gray, and Dr. Schief for such a rich set of discussions. I have a lot to take from this symposium, and I won't be able to summarize this because it will take us an additional 30 minutes or even an hour. But what I took from Glenda's presentation is she spoke beautifully about the target, the TPP for the vaccine that she's looking forward to have. And following that, Dr. Schief went on to talk to us about somatic hypermutation and how to activate a naive B cell into these super fecund plasma cells, which are going to secrete BNAbs. And you also talked about germline targeting vaccine design, all very interesting. Dr. Gilbert, it took us a lot through correlates of protection, and really this is where the field is going. We really need robust correlates of protection if we are going to succeed in this field. And last but not least, just to summarize, Dr. Shan, you talked about solutions to low immunogenicity of envelope antigens by DNA prime and protein boosting and high mutation rates of HIV, the solution, which you went to town to talk about polyvalent envelope antigens from diverse subtypes. So I've just picked like 100 of what our presenters talked about. What is left for me to say is to thank you and to hand over to Dr. Baden to close off the symposium and thank our sponsors for us. Dr. Baden.
No, thank you. Dr. Mgodi, and thank you for co-moderating with me. It's been terrific. And as you pointed out, this is incredibly encouraging symposium. HIV is a problem, a major problem that needs serious attention. And I am heartened to see the presentations and the multifaceted approaches that are going on for us to get to a vaccine, which is ultimately what we need to scale up globally. So I'd like to thank all the presenters again for terrific science, terrific talks, to the organizers for having this symposium on such an important issue during such a difficult time as we think about global health and HIV care. And especially thank my co-moderator, Dr. Mgodi, for co-running such a wonderful session. Thank you all for joining. There were upwards of 300 participants and even more questions, which again is just so heartening about the global dialogue. So thank you all, and we shall close the symposium now.
Thank you.